and definitely, people will continue joining the session, and they will become the part. So we can start.
Yeah, it's
Just
11.
give me 1 second. Okay.
Can I attempt sharing, Dr. Aparna, or shall I wait?
Just
Wait.
1 second. Yeah, let's get started. Very good morning, everybody, and welcome you all to the 31st webinar of the Plant Breeding webinar series. These webinars are being organized by the Foundation for Advanced Training in Plant Breeding on a monthly basis. And today we have Dr. Nataraja Karaba, who will talk about hologenomic insight, exploring the impact on physiology. The purpose of these webinars are to sensitize people to know and value the importance of plant breeding, and also to create an awareness on various aspects like technology integration, policy support, regulatory overview, farmers' comprehensions, and various other aspects required for inclusive development of crop improvement. Foundation for Advanced Training in Plant Breeding is a non-profit organization dedicated to enhancing capacity building in agriculture, particularly in the field of plant breeding. ATPBR, with its global reach and collaborative platform, stand at the forefront of fostering personal and institutional capabilities in alignment with current and future agriculture needs. We are committed to formulating strategies that contribute to effective crop improvement program and the development of human skills essential for ensuring sustainability in agriculture. The main objective of ATPBR is to develop sustainable agriculture R&D system to serve farmers, support in emerging entrepreneurs through advanced research and training, and assist seed industry to build market-driven competencies in human resource and make crop improvement an attractive and profitable business. ATPBR organizes professional trainings, monthly webinars, national and global conferences, and support publication. So far, we have organized 5 national conferences, 1 global conference, 27 professional training, 30 webinars, and reached more than 12,000 students and professionals representing agrarian community. Under the aegis of ATPBR, we have published 5 books, where books on market-driven plant breeding for practicing breeders, which we published with Springer, is our flagship publication. We have trained professionals from 66 organizations across India, Sri Lanka, Bangladesh, Brazil, Italy, Nepal, Afghanistan, Nigeria, Kenya, Vietnam, and Thailand. We have 12A ATG CSR certification, and we are eligible for conducting any CSR project for the industry. Now, I would like to take the privilege to introduce our moderator, Dr. N.K. Krishna Kumar. He is the former HOD Entomology IHR, former Director NBAIR, former DDG Horticulture, former Country Representative, Central and South Asia, Biodiversity International, and he is also Advisory Board member of ATPBR. Now, I request Krishna sir to please take the floor. Krishna sir, over to you.
Thank you, Aparna, and welcome Dr. Nataraja Karaba and all the members who are here. A very good morning to you. It's indeed a great pleasure for me to introduce Nataraj. In fact, we both are in the advisory role at Sericulture Biotechnology, where we had an excellent interaction for the last 3 years. And I truly consider him one of the intellectual brains throwing out a lot of basic research. Dr. Nataraja Karaba did his master's and PhD from the University of Agricultural Sciences, and he is now a professor at the Department of Crop Physiology, and he's head of the division and also heading the Central Instrumentation Facility at the University of Agricultural Sciences, Bangalore. Before coming to this position, he was for a very brief period at the Rubber Research Institute and also the Wood Sciences, which are located. And I'm very happy to share that he was a DST Boys Cast postdoctoral fellow at the Department of Molecular Cell Molecular, Cellular and Development Biology at Yale, a visiting scientist at the Plant Research International Wageningen University, Netherlands, University of Groningen, Netherlands, visiting professor at the University of Saskatchewan, Canada, and fellow at the in Germany. He is a recipient of Sir C.V. Raman Young Scientist Award from the Government of Karnataka, an INSA Teachers Award from the Indian National Science Academy. He serves as an expert member in various advisory panel in the Department of Science and Technology, and he served as he's also associated closely with the Associated Chief Editor of Plant Physiology Reports and is a serving member of the editorial board of Current Science. So that explains in a nutshell his accomplishments. His major research interest is understanding of the molecular basis of plant response to drought and other abiotic stresses. He comes from the laboratory of Professor Krishna Shastri and also the eminent physiologist with which the University of Agriculture Sciences is well known. And he has published more than 90 peer-reviewed papers in high-impact journals, right from PNAS, Trends in Plant Science, Trends in Ecology, Evolution, and to phytomedicine, fungal diversity, molecular breeding, plant biotechnology, and many others. 10 books, chapters, and 2 edited books as a continuation of major focus on abiotic stress. His team has initiated research to explore endophytes as a potential tool in modulating crop response to abiotic stresses and has made a significant uh, impact in the emerging areas. In fact, he was the chair when we had to discuss the recent advances in the breeding for the futuristic breeding on tomato, on which we organized a symposium. He headed the group with regard to the CRISPR-Cas and other advanced molecular technology as to how they can contribute to tomato improvement. And I was told that that was one of the finest sessions in the entire tomato conference. So we are looking forward to a great presentation, Dr. Nataraja Karaba. It's always a pleasure to listen to you. The floor is yours.
Thank you for the kind words, sir. I don't know whether I deserve all the things, appreciation, what you just said. All the work, what we are publishing is because of our students and team. I'm thankful to them. Okay, with this, let's get into the discussion, what we are planning today. Dr. Aparna, shall I share?
Yeah.
Please, sir, please.
Yeah. It might take a minute. I'm sorry for the delay. Is that okay?
Yes, sir. Yes.
Great. So again, thank you to Dr. Aparna and entire team there. Special thanks to Dr. Krishna Kumar for giving me this option to share my ideas to a heterogeneous group here. I know I'm not talking to hardcore scientists, although there are hardcore scientists there, but then there are people from different fields. So the idea here is to share the new emerging areas, which are very essential for overall crop improvement and in general, and crop physiology in particular, because I'm from the Department of Crop. Hello? Am I audible? No
Please,
problem?
please, sir, go ahead, please.
Yeah. Thank you.
Request everyone to mute their phone other than the speaker.
Okay. Thank you, sir. Thank you. So the idea of today's discussion is to prime you with the different fields. And our Department of Crop Physiology started many years ago by Professor Krishna Shastri, as Dr. Krishna Kumar just now mentioned, is one of the oldest department in the University of Agriculture Sciences, Bangalore. And our university is also well known for generating knowledge and networking. And we are very happy that we got an option to serve in this particular university and, of course, in the Department of Crop Physiology. So probably you will get more information later. Today, I thought I'll just focus on the issues which we are supposed to discuss: hologenomic insight, exploring the impact on crop physiology. Before that, I thought, since the group is diverse here, I will go with certain basic concepts. Then probably I'll start walking towards or making you to think about crop physiology and endophytes, their influence. And I will also tell how best it can be translated in the form of prospecting endophytes for trait activation, in non-host plants, because endophytes, we shall see later, are generally acclimated, adapted to particular conditions. So let's see how best we can do them and prospect them. And the possible mechanism of character or trait activation in the crop of our choice, we shall try to see quickly, not elaborate way. Then see whether we can have E-smart, endophyte smart crops and their possibility. That's what I thought. I know Krishna Kumar, like ecologist and entomologist, would be interested in holistic approach. So probably the talk which I'm going to do, hologenomic insight, would give some idea for all of us to work together and think together. So just for the beginners, all of us know that what we are is because of the gene what we carry with us. The phenotype is because of the genomic composition and genotype. That's we have been studying since many years. All the biologists know. And also there are some additional things, not just genotype can make the phenotype. You know that multiple things, the plus is very big and it is being added now. Many issues are being added, environmental effects and things like that. So now modern thinking started in a more broader way, that we have the hologenome, or the organisms which are contributing for the phenotype, as the additional thinking now, hologenomes. Hologenome is nothing but it's a study of collective genomes of an organisms, and of course its associated microorganisms. And this particular concept was there, and Adolf and Lin and others were actually proposed, and they were thinking the direction. But now we have a clarity to argue that the phenotype what we get both in animals as well as in plants is because of the hologenome, meaning genome of organisms which are associated with. So the hologenomic study is becoming very popular, and they do contribute on the overall plant physiology and plant biology and plant development process. So that's the reason we are working and talking today in these directions. In other words, hologenome comprises the total genomic components. And the total genomic components in the plant, say for example, you have chloroplast, mitochondrial genome as well as nuclear genome, plus the genome of the associated organisms which are living in the intercellular space, what we call it as apoplast. So that's what we mean at this point of time as a hologenome. In addition to that, this genome, that is hologenome, maybe host genome, also influenced by the associated microbes which are living outside the apoplast environment, as well as in the outside the plant body, that is the environment. So overall, we have a lot of influence on the genome, but at this point of time, let's remember that hologenome is basically a total genomic components of the host and associated microorganisms. That's what at this point of time we can remember. So what is the significance of such things? Be it in animals or in the plants, there's a lot of importance being given. And I will pick one example from the animal models, what people have been working since many years. So just to demonstrate the relevance of these organisms, people used twins, and see one person is fat, other person is lean in this example what I'm sharing. And they took microorganisms or microbiota, fecal microbiota in this case, and purified and fed to the genetically identical germ-free mice to see what type of phenotype you would get. As expected, the organisms which are from the fat person actually led to the obese phenotype mice, and you have a slim, lean person contributing for a slim phenotype. This very clearly demonstrates that the hologenome, or the organisms other than the host genome, plays a very important role with respect to phenotypic behavior. So that is the significance. So in other words, obesity and gut microflora in this example is influenced by the microorganisms. So the microorganisms therefore play a very crucial role in our behavior, activity, as well as phenotype what we are today. So I am not actually Nataraja. So I am basically because of the organism which are occupying in my body. And I am not talking, I'm talking as a hologenome along with all these microorganisms. I'm trying to share the idea to you. So our psychological behavior is also seems to be linked to the microbes, and probably Krishna Kumar can tell us more about that later. In other words, our body has large, the microorganisms. People argue that more than 10 times or more bacterial cells than human cells, and they occupy in different, tissue, even skin, inside, outside, because of their kind of habitat. acidic pH or oxygen enriched environment or low oxygen environment, things like that. So you have different organisms occupying different positions and talking to the host cells and communicating them to make sure that they live happily and also they transfer the benefit to the host. So significance of hologenome is being demonstrated everywhere now. In the same line, in our laboratory, some time ago, we started thinking about these organisms. And my colleague, Professor Uma Shankar, was working for the organisms or endophytes for a different purpose. So we started thinking of using them to activate the characters in the host plants. What are these organisms? We call them as endophytes. Endo means inside, which are living within the plant body. In the apoplast, apoplast is a huge area, which is a continuous network of cell wall as well as intercellular space within the plant body, connecting or, throughout the plant body from root to shoot. You have a continuous big network, and organisms occupy this place like an ocean. They do communicate and they derive different type of benefits, and also they give benefit to the host. So they interact with the host, perform a range of organisms and activities and functions. So the organism which occupy in the root may be different. In the stem, there could be different diversity, flowers, leaves, and you see a diversity with respect to their, the composition as well as abundance. So they are basically endosymbionts, and therefore we call them as endophytes, as a beneficial organisms. So this is what we have been talking about. And all these organisms somehow activate the characters in the plants and they impact on crop physiology as well as phenotype. All of us are aware that we have certain characters which are always there. We call them as constitutive characters. Some of them are induced whenever it is required, maybe drought period. We have increased accumulation of wax on the leaf surface, deep root system on the water limited conditions. These are all kind of induced characters. These sessile plants sense the conditions and activate. So now we started believing that these kind of genetic mechanisms or even epigenetic mechanisms are in fact directly or indirectly induced by the organisms outside as well as inside, which we have been calling as an endophyte. So endophytes have a tremendous role in modulating these both constitutive as well as induced traits in plants. So this is what we want. We want the characters, say for example, to improve drought tolerance, certain characters are to be induced upon mild drought stress. The root, which are sensing the water loss, top root, say for example, send the signal to the shoot to modify the phenotype to adjust the water saving mechanism. So similarly, these organisms can sense much faster and communicate to the host cell to make sure that they adjust their metabolic process. So that's what we have been talking and thinking. So in other words, original parent phenotype can be modified, altered with the help of certain microorganisms. We call them as, in this case, not organisms which are living outside. We have a huge microbiology department working on microorganisms. We are seeing from the lens of organisms which are living within the plant body, apoplast. So if you have in this article, authors argue that the microorganisms can, say you have A type of microorganisms can have one phenotype. If you have B type microorganisms, you can have another phenotype, which are quite different than the parent phenotype. That is the thinking with which people are arguing. So maybe deep root system or thicker leaf or a small leaf. So phenotype, therefore, seems to be influenced by the organisms which are present in the apoplast or endophytes. So am I okay? Hello?
Okay,
Yes, sir. Yes, sir.
Right, sir. Thank you so much. Sorry. So to prove this point, we thought one of our master students decided to erase the possible endophytes within the plant body and see whether we have some kind of compromised phenotype to demonstrate that they're essential for basic cellular plant physiological processes. So as usual, so we used 0.2% bavastin, which is a recommended dose, a systemic fungicide. So you can see when the plants are grown under kind of relatively disease or pathogen-free environment, you see the compromised growth of these seedlings, rice seedlings, when we treat with 0.2% bavastin. They don't kill the plant, but you see the difference in the growth. So what is happening here? We just try to erase some portion of the endophytes. That's all we attempted to do so. When we actually attempted to re-inoculate or put the endophytes back, you can see the enhanced growth although it is not on par with the original phenotype, but you see the better phenotype, which suggests that these endophytes are essential to maintain basic cellular process or physiological process, in this case, seedling vigor and seed growth. Not just that, we did look into the pattern of expression of certain basal genes, housekeeping genes, say, for example, BTF3, nuclear factor Y. I'm not going to the microbiology or molecular biology of this, but I just want to tell you that some of these basal housekeeping genes levels of expression were also downregulated when we erase the endophytes, arguing that they're essential. Endophytes are essential for basic cellular process, and we need to think about the treatment, what we have been doing, seed treatment with systemic fungicide. When we are able to grow the plants in a pathogen-free environment, I think it might come in the way of growth. So seed treatment may affect the seed endophyte, and that might probably affect the overall plant basic physiological processes of thinking, as demonstrated in this cartoon. In the presence of pathogen, well, you need seed treatment, but in the absence of pathogen, I feel, or we feel, there is a problem with respect to vigor and vigorous growth. So if you have huge endophyte load, and then we can probably have a better growth. And this kind of idea we published recently, and the seed treatment with systemic fungicide, we believe that it's time for us to review, and you have to think when to use, how to use, what dose to use. So they are essential for basic cellular process. The way people have demonstrated in animal models, maternal pool of microbiota is very important, and they probably contribute for unique metabolic process, or they may have unique genes which are essential for our development events. And people are arguing that mothers are responsible for transferring this microbiota to the offspring through different mechanisms. Infant growth and microbiome development within their plant body is influenced by the mothers. That is proven. There are many papers which are now being published, and we read them for our benefits to see whether such knowledge can be taken to plants as well. So, in other words, maternal gut microbiota are very important, especially during the pregnancy or early developmental stages, and that influences the overall behavior, health status of the organisms. So in case of animals, people also started arguing that dietary fibers or the microbiome beneficial system, what we have, I've been talking about endophytes, they do directly or indirectly influences maternal gut microbiota, and therefore they influence the phenotype of the organisms. So overall, we are connected. The endophytes, what I am talking, is also essential for the healthy growth and development of the organism, other organisms. So it's important for us to make sure that we retain these microorganisms and take to the next generation. I just want to make you to think about the domestication process, what we made many years ago, or 10 to 12,000 years ago. We started domestication of our crops and animals. And in this process, breeders did contribute significantly for overall growth and productivity. And they actually bred the varieties with the unique genes, cultivars with non-dispersing seeds, photoperiod insensitive, dwarf varieties, and many beneficial traits which are required for a large-scale commercial cultivation. In this process, we started probably losing them, or we ignored the hologenome component of the endophyte, which I've been talking so far. Say, for example, in this cartoon, I just want to explain that in wild-type species, say, for example, not improvised varieties, you have sufficient maternal tissue attached, and these maternal tissues carry significant portions of the microbiome. And when the seed germinates, you can transfer vertically to the next generation without much difficulties. In the domesticated modern cultivars, what we have been breeding now, and we have a mechanism of harvesting them, and shattering is not occurring, so that you can harvest them and thresh them in our controlled threshing yards. So we actually develop naked seeds. In this process, we lost maternal tissue. Therefore, we would have lost significant portions of the organisms which are helpful, apoplastic organisms which are helpful for our growth and development of our crops. So seed accessory structures, therefore, probably serve as a reservoir of the good microorganisms which are essential for the next generations. So this idea we published, and we are getting good feedback on this. The threshing yards where we thresh our seeds to get a naked seed probably is serving as a graveyard of maternal-born seed microbiome, which are very essential for the growth and development of our crops. And that's the reason our hologenome is not that active, and it is time for us to think in the directions as well. So people are now coming out with the novel ideas, latest papers about the vertical transmission of these type of organisms, beneficial microorganisms or endophytes, which are essential for improving plant fitness. And of course, microhabitat also influences the seed microbiome, and overall that actually contributes for the phenotype. And it is time for us to think in the direction to make sure that we preserve somehow the seed accessory structures so that we can probably retain some of these organisms or endophytes, which are now being considered as vertically transmitted microbiota. So I want to impress you with this one of the latest papers which I read a couple of weeks ago, appeared in Nature Ecology and Evolution. So beautiful experiments they did. In this cartoon, if you are able to see it properly, I have different bacteria, or they have shown different bacteria in different color combination and the same color combination of clonal plants. So these endophytes or bacteria in this case are asked to colonize independently here, and the induction of diverse phenotype you can see here, although we have a clonal plants with a different type of phenotype. That's what they are trying to demonstrate in this paper. So in this case, they exposed the volatile from the microorganisms. They did a beautiful experiment to make sure that volatiles are exposed to these plants from different bacteria. The interesting thing, what I want to say is that it has huge impact on the phenotype. Look at the leaf morphology here. F23, F37, F31, and F12. These are different organisms. And I'm really impressed to see this phenotype. Totally, they change the phenotype. Not just that, this microbe-induced phenotypic variation, in fact, led to significant increase in the biomass also. Well, when you change the canopy architecture, it's possible. Better photosynthesis and better translocation of metabolites possible. And that would have contributed for the biomass. Agreed. But see, a small kind of volatile compound from the hologenome, or I would say endophytes, is capable of doing such huge changes in the phenotype, says that we need to think aggressively towards this concept and see how best we can explore and use it for our crop biology and manipulating the traits. From this context, we started a few years ago exploring, exploiting, or prospecting endophytes to activate the characters in the plants contributing for drought tolerance. So we thought we can use endophyte as an elicitor. We have now a nice program on prospecting endophytes. They are called trait-specific endophytes, capable of activating characters. So our goal is every endophyte will not do everything, so we need to prospect, mine them, look for the good characters without having any kind of bad effect on our host plant. So trait activation in crop was the goal. So in this cartoon or the photograph, what I'm showing is you have the endophyte being screened for salinity tolerance. So you have endophytes capable of growing at 2.5 molar NaCl. That's great. So when we isolated these endophytes over 400 plus, we started playing one by one, and some of them are able to colonize in the host of our choice. Say, for example, rice. They are isolated from the harsh environment or the plants growing in a harsh environmental conditions. They are stress adapted. We screen them under laboratory condition, ask them to colonize non-host plants. In this example is rice, and you could see the significant improvement in thermotolerance, which Dr. Krishna Kumar was just now mentioning before in the talk, that there is kind of hot winds and those kind of situation probably can be manipulated to some extent with the help of these type of organisms. I just want to show some pictures, not in the details, because some of these are still emerging stories in our lab. Say, for example, NaCl, one organism P10, you see the seedling vigor compared to only when you challenge with NaCl. And like that, many more. This is in case of horticulture crops, both field crops, horticulture crops, dicots, monocots. We have been screening them, asking them to see whether they can activate the characters in our laboratory conditions or greenhouse conditions. As we see, sometimes we look at the growth, sometimes we look at the characters which are essential during drought or salinity stress tolerance. So some of our masters as well as PhD students, we have an army of researchers in our labs. So they help us to think as well as do the experiments. So they demonstrated that they are capable of enhancing root growth and development, some of these endophytes. Well, that's important for us. We call them as water mining characters under drought situations. We need such plants, induced characters. So root length, shoot length, as well as the overall seedling vigor has been enhanced whenever we challenge under laboratory condition with desiccation stress like PG, polyethylene glycol induced stress, simulating the drought stress in the field conditions. So I just want to impress you that endophytes, what I call as the beneficial organisms, or you can just think from the angle of hologenome, contributes for the phenotype. This particular paper, which was published a couple of years ago, is very impressive for most of the people who are working in this domain. So, for example, when you have endophytes colonized, you can improve overall growth and development. In this paper, we isolated endophyte from a salt-tolerant Pokali rice variety, asked the endophytes entry into a salt-sensitive IR64. Just entry itself enhances its growth and development under laboratory conditions, greenhouse conditions. In the absence of endophytes, you don't see that robust growth under laboratory condition, but you do see a significant growth. You put a salt, 4 decisimum, 6 decisimum, we have series of different concentration of salt stress imposed. You can see no compromised growth at all. On the other hand, salt press endophyte minus, you see a tip burning, typical symptom of physiological disorders. See, it's very interesting. What does it mean? The IR64 has entire gene pool required to activate the resistant mechanism or tolerance mechanism for a salt stress, but it was waiting for somebody to activate the characters. So breeders have already probably put in all the pathways or the genes which are essential for tolerance characters. So this is very impressive. And now it's interesting for us to ask the question, what's happening? Why this phenotype is, not expressed in the absence of endophytes, which we are asking. So the Pokali rice variety or similar type of rice varieties, therefore, can be a powerful source of endophytes or apoplastic organisms required for activating the characters in the sensitive varieties. Is the thinking of ours, and we are doing some experiment. And also, we are having some leads to say that they are vertically transmitted, and probably we'll have new varieties being generated through these kind of approaches as well. So I have a series of experiments with the different pulses, dichots, monocot system, and the image what you are seeing right now is being done under a project supported by ICR under NICRA. I'm thankful to Krishna Kumar who encouraged us to have this project. And we have early flowering phenotype in case of pigeon pea and significant improvement in the biomass as well as yield under greenhouse conditions, and field experiments are going on, and we are getting very impressive data there as well. So similarly, we have tomato. Since there are people from the industry, I thought I would pick up these kind of examples. And tomato also, we have profuse vegetative growth, but we are trying to see why there is not much growth with respect to tomato fruits. We are not having significant increase in the yield under greenhouse conditions. So we are taking this experiment to the field out of protected cultivation. But overall, we have now endophytes capable of activating the characters, both growth as well as stress resilience. As I mentioned, it's not just in universities like us, trying to see and leave it. So we are also asking the question, what are the possible mechanisms by which these endophytes communicate to my host and activate the characters? So this is from the rice experiment. And we have some kind of, I'm not going to discuss in detail. We have some kind of indication from transcriptome analysis, global transcriptome or RNA-seq analysis to say that there are calcium signaling events. So early signaling lead to activation of certain specific gene pool or signaling pathways, which are associated with the salt, so-called tolerance or detoxification events. So in other words, the Pokali rice endophytes activate calcium signaling mechanisms. So calcium signaling is a complex process. So we will be mining more, and I'm fortunate to have a collaboration in Germany. So they're also helping us to look into these aspects in a collaborative manner using targeted metabolome analysis as well. So sometimes playing with the croplets, it's difficult to address the critical issues. If you identify the signaling molecules or the metabolites, it would be easy for us to manipulate in a targeted way. So from that context, we started playing with Arabidopsis thaliana, a model system. And this model plant is being used for multiple purposes in our biology, plant biology and plant physiology. See, one of the organisms which we are playing with has profound effects on phenotypic differences. This is without endophyte, with endophyte, and huge difference in the root growth also. And now we are seeing from the angle of water use efficiency, water mining and water character, as well as nutrients efficiency using this model system. And we have some mechanism to collect epiplastic fluid and to see what are the communication possible mechanisms. And that might give us more ideas, and probably we can manipulate certain characters or activate the host genome in the absence of hologenome in case if we land on the good molecules. So that's the thinking with which we are working. And academic research is sometimes exploratory. We are trying to explore all angle to get some idea what is happening with respect to communication. So this particular field, what I mentioned, beneficial apoplast, microbes, is very interesting. And a couple of days ago, I was just mining some information for this talk. I came to know that over 1,553 papers published last year. This year itself, 311 papers. Just the hit endophyte gave us. What does it mean? It means people are very much interested. It is highly emerging field, and probably we might get a kind of translatable ideas which can be translated to the field without any delay. That's the reason people are working and thinking in the directions. So I have some concepts, or we have been working on certain concepts as a crop physiologist, how it enhances growth. If there is an increment in growth, means there is enhanced carbon. So carbon must be through the process of photosynthesis. We have certain concepts and ideas. Well, this is not the forum to discuss all these ideas, but if anybody is interested, we are keen to discuss. And endophytes, they occupy the apoplast, as I mentioned. They might enrich carbon dioxide in this microclimate area, and that might enhance photosynthesis. We have an enzyme called RUBP carboxylase oxygenase, which does carboxylation as well as oxygenation process. So when we enrich CO2 and decrease oxygen concentration because of microbial activity, and there is a possibility of enhanced photosynthesis. This is one of the thinking which we are working with. And because most of the plants enriched with the endophytes have high rates of photosynthesis, and we also have a concept to argue that endophytes trigger source activity, and they serve as an additional sink in the apoplast. Therefore, there is no end product inhibition on the photosynthesis, and this end product inhibition can minimize photosynthetic limitation. That's an interesting thing which we published recently in Trends in Plant Science, and ultimately we have some kind of arguments to the issues to demonstrate that the endophytes minimize photosynthetic limitation. They enhance photosynthesis. They retain photosynthesis in a day for a longer period. Therefore, there is enhanced biomass, and then you have more source activity, and the source, that is carbohydrates or photosynthetes or distributed zinc. Therefore, there is enhanced growth and development. It's a thinking, and we are generating data in that directions as well. So in other words, it's nice, beneficial. Apoplastic microorganisms are doing great to the plant system. They can adapt to the changing environment much faster than the complex host like plants, and therefore they can collaterally offer such adaptation to the host, and they can serve as an integral component of the plant. Probably they can be transported vertically if you retain some maternal tissue, and you can evolve a new plant types without much so-called complexities. So the phenotype, which is the combination of G into E, everybody talks about it. It is genotype as well as interaction, is now viewed from a different angle with an E as a residual error or additional components to make sure that the complex phenotype is understood in a more holistic way, and the model of G into E is being complicated with new quantitative features, or you have a host genome, endophytes, that is hologenome, and their interactions. So you have a model. People are arguing that there is a GEM model, GE and microbe associated model, and of course environmental interaction is must. All of them put together governs the phenotype. So you have hologenome playing a very crucial role in addition to the host genome. So all these models help us to think and give an option handle to manipulate. So host genome, microbe genome, and of course interaction with the environment, other microorganisms shape the phenotype. So we have to think in that angle And hologenomic insight is very essential. Exploring the impact on crop biology and crop physiology is very, very crucial. And remember that there are specificities. All organisms cannot occupy in all zones, all type of tissues, as demonstrated in animal model. Even in the plants, we have different types of organisms contributing for the beneficial activity in different tissue types. So you may have different types of organisms in the root, collar zones, fruits, or even in the leaf, which might contribute for the growth as well as quality of the fruits in case of vegetables and fruits, if you think from that angle. So endophytic microbiome plays a very, very crucial role in overall crop growth and development as well as crop physiology. So there are now interesting stories being or hypotheses being proposed. Say, for example, if you have ideal microorganisms, endophytes, they all help the plant to be a kind of happy plant, and microbes from fruits and vegetables enter to the human system. They also make sure that human system is also with ideal endophytes, or sorry, microorganisms, not endophytes there. And we also know that we are unable to synthesize all the essential amino acids. Some of these essential amino acids are coming from either from food or microorganisms. And therefore, the ideal microorganisms contribute for the ideal growth, good health. And if you have a poor endophytes or the hologenome in the plant, probably they may contribute for a fragile health because they are unable to contribute for the healthy growth within the plant, our system or animals or plant system. So you may have some kind of compromised growth and development because of maybe vitamin deficiency, minerals, or essential metabolite production. So that is the holistic thinking people are thinking. So the entire thing should be seen as a system, not in isolation. Breeding is important. Please make sure that we also look at not just genome, hologenome. Improvement, therefore, means a holistic approach, not a focused one directions. So that's the thinking of everybody now. So therefore, people are now moving in a big way that Earth Hologenome Initiative. It's basically different activity, but we are, as academician, interested in science, basically to generate and analyze hologenomic data to answer the basic questions, fundamental questions, which might have huge impact in future crop production as well as human health. So I am just having a kind of vision of having e-smart crop plants, that is endophyte enriched, hologenome enriched crop plant, capable of enhancing, say, from the angle of drought tolerance, which we are working in our lab, enhancing root activity, root growth and development, or water mining characters. There are examples already. And capable of maintaining good cell level tolerance in harsh environmental conditions, sudden temperature rise, or there is an issue with respect to oxidative stress. They may maintain the cellular metabolic activity, they maintain membrane stability. There could be endophytes, there are indications already capable of maintaining basic cellular process. We call them as cell level tolerance, as well as closing stomata, producing more amount of waxes to conserve water, reflect radiation. So we call them as water conservation mechanism, making the stomata hypersensitive, efficient stomatal movement. We are looking for the organisms which can do this. And if you combine all these organisms, probably you can have a smart plants helping us, and they have potential to bring rapid evolution changes. As I mentioned, they adapt much faster than other organisms. Therefore, they can adapt to the changing environment much faster. So I believe hologenome or endophytes have a potential to bring evolutionary changes in the field of agriculture. All of us, industry as well as academicians, should work together, we should join hand to make sure that we have a healthy plant, incorporate our knowledge, both field as well as lab knowledge, to contribute to sustainable farming system. Ultimately, our goal is to have sustained yield and quality. Of course, food and nutrition security is very important. I know that Krishna Kumar keeps talking about nutritional security and eco-friendly manner, sustainable farming system. And we have a small idea from this angle of hologenomic insight, which not only improve the growth, it can maintain hepatic stress tolerance, plant health, as well as defense. So that's what I just wanted to share. Before I conclude, I want to recall the help of all the people who are working with us directly or indirectly: Professor Umashankar, Ralph, Suryanarayanan, Theo from the Groningen University, and Rogottama from Purdue University, Karen Tanino from Saskatchewan University, Canada, Paranjit Kurana from University of Delhi South Campus, and Padmaja, my collaborator in the project, supported by NICRA. And all the work what we are doing is because of our students and research associate and Pallavi, Rachana, Abhishek, Chetana, Kiran, Danyalakshmi, Mansa, Hemapriya, Aisha, Akshata, Aishwarya. The list goes on. I only picked up some names because I did not use others' data, but they did contribute for the growth and development of our ideas here. And special thanks to the team here. So ATPBR team, Krishna Kumar, Kiku and Aparna for giving me the options to share some of our lab activities and also activate your thinking on hologenomic insight. Thank you so much.
Thank
And
you.
special thanks to entire funding agency. I have NHEP funding program under NICRA, DBT and ICR and World Bank funded some of the activities on endophytes. Thank you. With this, I conclude my talk.
Thank you. Thank you. For a wonderful, very fundamental but profoundly the one which is futuristic, one which is trying to understand. It is said that an expert is one who knows more and more about less and less. And I think this is a classical example of trying to know more and more about things we otherwise do not even probe further. I would like in the queue, first I thought Dr. Tiku had raised his hand, then I request Navalappa to, after Tiku, to seek clarification. So Dr. Tiku, please go ahead.
Thank you. Number one, excellent talk. I mean, you have removed a lot of my cobwebs on this. So I really appreciate, and it's so precise, finished in 45 minutes, but very clear. Because sometimes, we can keep on talking, but very, very clear, and a lot of cobwebs have been removed. My congratulations. So this really is a topic that, of late we have been hearing quite a lot, and I think you are an expert on that. So I just wanted to know epigenetics and this. How would you relate them? Is it similar or same thing, or because epigenetics is also something outside of the nuclear genome, right? So how would you, because these days a lot of people are talking about epigenetics being involved in the breakdown of virus resistance, say in okra. When we go to different locations, same genome, but then you find it's susceptible. So your thoughts on that? Thank you.
Yeah, thank you, sir. Maybe Dr. Krishna Kumar, sir, can also add some points later because we keep debating on this issue.
Yeah.
You are very correct. Epigenetic is the concept which we are arguing here, which cannot be explained by the Mendelian pattern of inheritance.
Yes.
Right? Say, for example, I have applied the rice variety. I grow them under puddled condition.
Yeah.
And all those seeds to grow under upland condition, they do not behave well. That means there are certain epigenetic changes happening on the DNA.
Exactly.
Be methylation marks, or we have some concepts called modification of the DNA, but they do not change the gene structure, but then their activity is altered. So that's what we mean epigenetic mechanisms. So maybe it is possible with the virus. And in our endophytes also we see when we ask the endophytes to grow continuously under laboratory conditions and they forget to activate the characters. So we can put some kind of methylation inhibitors and activate that process again. So in other words, DNA methylation is one of the indication of epigenetic modification that is happening.
Yes.
I hope I gave some idea and maybe others can add on to it. Thank you.
Thank you. I just want to add to that you talked about rice. So it's clear that means that you can't just take out the rice varieties that are growing in puddled conditions and put them under DSR. So you may have to have the right combination of the varieties and whatever else is required to get the right combination, right?
Yes, sir. You are right. You are very correct. It's not just presence of the gene, even signals. Epigenetic modification also matters during their growth conditions.
Thank you.
Dr. Nataraja Karaba, you please also share your phone number. There is one Dr. Dilip Verma. He says, can we get the brief biodata of Dr.
Yeah, I think Aparna can send it to
Aparna can send it to
Dr.
Dilip
Dilip,
Verma.
yeah, because
Yes, sir.
she had shared it in one of these things. So I think we can do that.
Sure, sir. Thank you.
Sure, sir, sure.
One more person had raised his hand with regard to asking. Please go ahead. Anybody one by one? Any questions?
They must have raised the hand to say just
No.
cheers
Okay.
for his talk.
Dr. Nataraja Karaba, I was couple of times I have been following on the microbiome associated with the stigma of the flowers, which I thought I'll share it also here. That will be more critical because one of my student is working on the microbiome associated with the stigma of the flowers.
Yes, sir.
And it is very beautifully coming that the very often that we see as heat tolerance or either tolerance or lack of fruit set is often influenced by the microbiome that is on the surface of the stigma.
Right.
And what was more beautiful was the microbiome very often communicates with the host or the others through the volatiles.
Yeah.
And I think the volatiles, see, there is a very beautiful biology and chemistry playing because the communication is through volatiles
Yes.
and the organism. So it happens. And they further went on to say how the pollinators can bring microorganisms from outside and carry the microbiome from one to another, thereby increasing the fitness, also adding to the value of pollination. And excellent work is being done in this direction.
Yes,
I think
sir.
I also sent to Dr. Tikku and you now in your WhatsApp one more wonderful publication on the microbiome associated.
Thank you, sir.
And
Thank you. That's a good thing.
what I am worried is how much are we really focusing as much attention on the volatiles coming out of the microbiome as much as the microbiome itself?
Yeah. Can I, sir?
Yeah.
So that's the thing. You are very correct. Sometimes this paper which I was citing shows that it is not just the organism or direct metabolite communication or the apoplast signals. What we were looking at calcium signal, it is a volatile that neighboring plant also have the same impact. Vicinity or close association with the cell may or may not be required always. Neighboring plant can have the phenotype thrown out. That means your point is well taken, sir. All kind of things, including volatiles, play a very crucial role in communication as well as altering the phenotype.
In order to keep the discussion going, because many others are not, what is the microbiome profile within a species between cultivated plant and the crop wild relatives?
Hello, Dr. Dilip has raised a hand. So
Vineet, please go ahead.
Sir, just in one of the slide, I go through that my hologenome, and another it was hologemon. Is it only by mistake or is
Possible, sir. Sorry. It is hologenome. Genome. Sorry.
Typo.
okay? In one of the slide I had seen, then I thought that
Very
it
sorry,
should
sir.
be wrong.
Thank you so much. So you are a good student.
So nice, so nice.
Sometimes it's
Very sorry. Yeah, typo. Sorry.
typo. Yeah.
Could you answer that question, Dr. Nataraja Karaba? Has any such work been
Yes,
carried
sir.
out?
One of our students working with Professor Umashankar attempted that, and she made comparative analysis of cultivated versus kind of wild accessions of finger millet. We do see a diversity variations with respect to the diversity of microorganisms, but at this point of time, we don't see that significant change. Maybe their cultivation habit and all that matters. Outside fellow might influence inside fellow.
Okay.
So that might complicate the analysis. That's what it looks like.
Dr. Tikku?
Yeah, I was reminded of, how does this differ from allelopathy? We used to talk about allelopathy a lot. I'm talking of
Yeah.
we talked about how do they differ from this microbiome? Isn't that also similar or same?
Okay. Allelopathy, what we mean, sir, from the angle of plant, a plant releases certain compounds which inhibits the growth of other plant. Allelopathic effect. So basically, it's a suppression of growth for their evolutionary fitness. So here, what we are looking at is organisms which are mutually benefited, like symbiotic association. That's what we are seeing.
So
At some stage, they may not like the association, but we believe they live together because they like each other. That's the idea there.
Okay, I thought allelopathic also meant symbiotic relationship between the two, or no?
Yes, no, sir. That's what I would say.
Okay.
It depends upon the conditions. Yeah.
Okay.
Yeah.
Okay.
We welcome Dr. Basuraj Khadi. Khadi, do you have anything to ask? Anybody else? We have quite a good number of participants, and this. Do you think, Dr. Nataraja Karaba, now it is wise, before we release a variety or identify something as very, because what I'm really beginning is now plant breeders are looking into also the holistic compositions of the microbiome before they would like it to go to seed.
Yeah.
So that kind of an approach is
Namaskar.
coming to plant breeding, and your say on this.
I was a student at university.
So that's what I was just hinting, sir, in my talk. So as you rightly, beautifully mentioned, it's a kind of breeders would have already packed most of the genes which are required for diverse pathways, water or nutrient use efficiency, etc. But they are quiet. But if I have some activators like this, like the beneficial organisms or endophytes, what I've been talking, and you can activate that pathway and make sure that plant is capable of exhibiting those traits. So in other words, it is important for us to first enrich the hologenome and also simultaneously make sure that breeders should think about hologenome, not just host genome.
Wonderful.
So that's the idea, sir. That's, I think it's a correct way of seeing it for holistic crop improvement.
Dr. Khadi?
Thank you. Thank you. Thank you, Uncle.
Right.
Anybody else?
Yeah, I have because
Yeah, please.
this the breeders are developing new hybrids, new varieties, but that's why I think the last 10 years, one of the things that has happened is without multi-location testing, we don't release anything new for any zone, and that has been the reason why All India coordinated testing is also done. Now, how do you add this component into that All India level testing for both private sector and public sector? There should be some learning from this. So how do we, kind of integrate it into that function? Because that function is now an important function in almost every serious seed company. We call it product development. Some call it technology development.
Yes, sir. So I am just thinking wild, but then I say the breeding program should go in a microbial way. You have your program. At the same time, you should also have a library of microorganisms, germplasm of microorganisms, and you have to mix and match and make sure that you get more benefit than what you are getting through breeding program. So it should go with the microbial way is the thinking, sir. And some of the things are vertically transmitted, meaning seed to seed. It is being seen in wheat and rice, what we are doing under NICRA project now. But then some of them are not vertically transmitted. So we don't, probably in pulses, it is difficult to make the endophytes to go from seed to seed because of maternal tissue is not attached with most often. So that cases, every time you have to make sure that you enrich the microbiome and give to the companies, translation. And microbial way is essential. You should have a microbial pool or a germplasm for seed as well as for microbes. That's the thinking of mine, sir.
I think this needs some discussion and, so that a plan can be put into place. So I will be following you.
Sure, sir. Sure.
Yeah.
Sure.
Maybe I should come to Bangalore for a couple of days.
Please,
Sure.
my pleasure,
Please
sir. My pleasure. And I repeat again, I am not working on microorganism. I am working on the endophytes, the organisms which live within the plant body in the upper class regions.
Excellent.
Microbiology department is huge. They have a lot of rich knowledge in that domain. I'm not seeing from that lens. It is endophytes which are living within the plant body.
Yeah. Okay. Thank you.
Thank you very much, sir.
Anybody else? Please, we request others also to raise questions, because these are all cutting edge research and we need to students and others to really participate.
There was a message. Ruchi
Dr. Ruchi Parida, how microbiome activates defense mechanism against multiple stress?
Yes. Same thing.
Hello.
Dr. Nataraj, you can answer.
Yeah.
How
So-
microbiome activates defense mechanisms against multiple stresses?
Correct. So I have used in one of my slides, the stress response alters the cellular level machinery or cellular activity. So be it salinity stress or a drought stress or a high temperature stress, there will be changes to the membrane, cell membrane. So you have membrane stability, unstable membrane. So lipids and other protein structures are getting altered. So if you are able to maintain membrane stability by some mechanism, maybe quenching reactive oxygen species or any other mechanism, then you can improve stress tolerance by altering the cellular level tolerance to drought, salinity, as well as high temperature. That's the thing. So if any organism, say endophyte, what I'm playing with, capable of maintaining cell level tolerance, probably is also capable of imparting multiple stress tolerance. That is the thinking with which we are working. Although one organisms cannot do everything, but if you attack the basic processes and look for the organism capable of maintaining basic physiological processes, probably it is possible you can have one organism doing the major jobs. That's the thinking of ours. Thank you. Good question.
Thank you.
Anyone who wants to raise a question?
Sir, I am Maheshwarappa, sir.
Please, Maheshwarappa, go ahead.
Nataraj,
Namaste.
Dr. Nataraj, really, it is a very good talk, and I compliment for the talk and your work also. I think as Tikku sir told and Krishna Kumar sir also told, I think there is lot of scope for doing this type of work in our horticultural crops, I can say, for mainly
Yes.
for the any, as you told, when you look into the breeding angle, because only few works have been done with respect to the resistance for a particular pest or disease only. Some here and there work has been done, but it is not systematically done as you have done it. I think I request you to just start working on some of the important horticulture crops by looking into your experience on a certain aspect, the role of these endophytes as a whole thing. I think. Thank you very much. Thank you very much.
I think you should call for a short brainstorming session at your.
Yeah, definitely, sir. Definitely, sir.
We'll
I will
do,
be inviting.
do with
Yeah.
Maheshwarappa.
Yes, sir.
Maheshwarappa, what I would advise you is, let one of your physiologists start a program on this, and with the active help of Dr. Nataraja Karaba,
Yes, sir.
so that
Yes, sir.
what happens is your students and your staff, taking the help of Nataraja Karaba, could synthesize a lot more information with regard to many of these things.
Definitely, sir. Because
Please
we
go
have a
ahead,
camp.
because there is also a limitation how much UAS can expand in horticulture. But
Yeah,
if
yeah.
you start, he will be certainly, hand holding can be done by Nataraj Kadaba.
Yeah,
And
definitely.
a synthesis of two universities will always be good.
Yeah, definitely, sir. Because we have college in Bangalore only. So
Yeah.
there I will request our colleagues to have interaction, and we will work together, sir. Thank you, Karapa.
No, I call it the other PPP. That is not happening. Public partnership is not happening.
That also we'll see, sir. We have to call some public entrepreneurs. Then we can have.
Dr. Maheshwarappa is the director of research, and certainly the research, please take it forward. There are
Yeah,
a
we'll do, sir.
number of problems.
You can take the lead.
Yeah, sir. We'll do, sir. Thank you, sir.
Dr.
Any
Maheshwar,
work-
thank you for the kind words.
Thank you.
As you mentioned, we initially started with a different monocot system, pulses and all that during our World Bank funded project.
Okay.
Now I have limited grants, and therefore I'm just playing with the tomato system.
Okay.
And there are two things we are doing. One, the way I said, whether it can do something with respect to growth and development under open environment. Second thing, I'm also using tomato as a model system to harvest apoplast fluid, that is outside the cell wall. There are certain, apoplast fluid. So we are trying to collect those apoplast fluid and ask the question, what are the metabolites or what are the signals that are there in the presence of endophytes, in the absence of endophytes? That's the thinking. So thank you. And maybe we will join together. Good advice by Dr. Krishna Kumar also.
Yeah, definitely.
Thank you so much for your kind words. Yeah.
Thank you. Thank you.
Thank you.
Anyone else? We like questions to come. That's how the science. Sir, how can we differentiate soil-borne pathogen and endophyte? Nataraj Kadaba?
Yeah. So the endophytes, again, I repeat, we are not at this point of time talking about the organisms which are outside the plant system. We are talking about organisms which are within the plant body, within the apoplast. Not like pathogen. Pathogen actually degrade the cell wall and they have some lytic enzymes or they destroy the system. So we are not looking at those organisms. We are actually at this point of time looking at the organisms which are living within the apoplast and helping in some form without any disease or bad effects on the growth and development of the plant. So that's what we call, and they calculate basic plant physiological process. Therefore, we call them as hologenomic components. Host genome plus microbe genome. I hope I addressed the question. Thank you.
I think his question was different. Can you read that question again? Or?
Sir, how can we differentiate between soil-borne endophyte and pathogen? Sorry, sir, my question is second one. That is,
So this is the question. Soil-borne endophyte.
Between soil-borne endophyte and pathogen.
So if it is soil-borne, sir, they are not basically called as endophyte. They may be there as a spores, or they are waiting for the host to enter inside. So it's just like, seed or a spore. The endophytes, what we mean is they should live within the plant body and they contribute for their growth within the plant body. So they are not germinating and living there. Even if it is there, it's for a transient survival. So it is maybe there because they would have been there because the debris are there. That's how it is.
I would also like to add here.
Can I say something?
Yeah, please, sir.
Just for the knowledge of everybody, I was reading a very long paper, and also it's a video somewhere on this microbiome in human beings. Cornell University, some doctors from their medical school, the doctor said that now they don't want to say that 4 people have a headache, all of them take crocin and you will be all right. We have to study their microbiome and then get the right medicine for them. And this is now getting into the medical system, that they have algorithm data on the microbiome of all the people. So they have health data. So when you are having symptoms, they try to see your microbiome and, kind of correlate it like we do genomic sequencing. So they do the sequence correlation and then prescribe the right medicine. So this is where they are reaching, and I hope in India also this is happening or not. So this is where also your lecture kind of, rang a lot of bells to me. So I've been reading that for the last 1.5 years. Pretty, very, very, very nice videos it's all on YouTube. One can find the Cornell University. I forgot the name of the doctor.
That's right, sir. That's right. That's the reason this Earth hologenome concept started, holobiome started.
Yeah.
So everything is being sequenced. You are very correct. It is being practiced even in medicine in developed countries. You are right.
Thank you.
I would also like to add here, even in viruses, it has been for a very long time known that there are helper viruses. There are helper components in a viruses.
Yes.
Certain viruses like Bryce Tungro get expressed only when 2 viruses just make a uniform kind of balance within the plant system to create the Tungro viruses. So what we are seeing is, I'm not telling that the hologenome of virus, but they may have their own very intricate mechanisms by which they can one way or the other way exploit different kinds of what I don't know. So I'm sure that it is a very fascinating field. The more we begin to know, the more we understand. I think it appears to be more beautiful and yet complex. But my one small question is, for example, in many plants, the salinity tolerance, water stress tolerance, and many others go hand in hand. So are there a cluster of microbiome that you can associate with, for example, drought versus submergence, or those kinds of things? Is there any cluster? Second one is it possible in some way under this situation externally, we can help the microbiome to express better?
That's a good question, sir. That's what people are asking now. And if a organism attacking a basic physiological process, then it is possible. Probably you'll have multiple, resistance shown. But if they are highly specific, that we will know only when we understand them, then we may have to have a consortium of organisms. And again, there is a question, when 2 genomes, microbe genome, work together, there could be so many other complications, compatibility related issues. So clustering or consortium model, everything is being addressed. And before that, people are now asking, what is the communication mechanism which is happening between microbes and the host, in this case, plant system. And once you understand that, then we have all options to combine them, make sure that they don't become pathogenic later, or they don't turn on their activity and turn off their activity. So that's the line of thinking, and you are very correct, sir. There could be a way by which we can have a consortia and treat for a XYZ problems.
Thank you very much. That's Dr. Khadi, you would like to ask something?
Thank you, sir. No, I don't have.
Okay. So, Aparna, are you there? Aparna had told me that she would have to leave, and she told me to summarize. So shall I give my final wrap up? Is it okay, Dr. TK?
Yeah.
So
Yes.
I profusely thank on behalf of HPBR the wonderful lecture. I think after a long time, I'm hearing something which is very stimulating to the brain, and lot of unknowns are being, made clear with regard to the aspects that were dealt. For example, the Dr. Nataraja Karaba started with, many of us really didn't have a very specific understanding of what is hologenomics, and he very clearly said how it is important for crop physiology, as well as then through physiologically how the endophytes or the hologenomic analysis insights can help us in understanding various problems that are otherwise not very clearly said. He also said that the significance, that is, these organisms are those, there is a host genome which is present in the nuclear, mitochondria and chloroplast, plus other microbiota which is contributing, which he defined as the hologenomic, this thing, significant is the role of the presence of them in the apoplast. Apoplast is very clearly said are those that are outside the plasma membrane and the cell wall, and which lot of communications goes through. He very specifically took the example as to how application of a fungicide can have a detrimental effect on the endophytes, and thereby restrict in an otherwise a plant where there is no pathogen, and unnecessary application of any fungicide or a pesticide, how detrimental it could be. This is very significant from a practical perspective, because in the name of prophylactic spray, if you go on dumping very harmful, then you are playing with the microbiome that are associated, or hologenomic composition that are associated with the plants, and thereby the plant will not be able to express its full potentiality. He also made a very wonderful difference between the genotype and the phenotype, and how these two things are also to a great extent influenced by the hologenomic compositions. And very happy to know that it can flow through the mother into the offspring. Maternal gut microbiota is very important. He briefly touched upon the animal and the role of the dietary fiber and its impact on the endophytic composition, and also briefly touched upon how the domestication of the plants has taken place. And while we have been able to focus on certain traits, how the loss of certain a very precious part of the hologenome could actually make that plant. Probably now we should ask the question, why is it that many of our hybrids and varieties that we are releasing, by default, they are not as resistant to pest and diseases as they were? Somewhere, whether we have lost some of the very useful genes through the hologenome. And I was also very keen to know about the volatiles, since in honeybees and others, the volatiles of the stigma composition have also been very clearly indicated. I don't know, Nataraja Karaba, I passed on that paper to you. If it is not, I'll pass it on to you. Okay. And the specificity of the endophytes with regard to fitness and the non-host fitness. And he also talked about how the rice, Pokali rice, and some of the microbiome that is associated with Pokali rice when it was transferred to IR64 enhanced the growth and the salt tolerance, which was otherwise not visibly seen. I think this is what I see the very practical aspect of, and this is something which you should see for problems such as bacterial wilt and many of the that are associated with it. I think there is a more to do, and he gave through some of the very wonderful laboratory example on the nitrogen and water use efficiency too, how these things can play this thing, and they can enrich photosynthesis, enhance the biomass, and also the growth and development. Of course, he also briefly touched upon the concept of G into E is now expanded into the host genome, hologenome, then their own interactions in addition to the environment. See, G into E appears to be much more complex than otherwise normally visualized, and but there are specifications as to where they can be. That is talked about the specificity, and very happy to see the concept of e-smart plant where endophytes or the other things are also recalibrated with respect to the overall fitness, and the also some of the responses that you gave to the questions were wonderful, with from epigenetics to many of the others. I think overall everyone who participated tremendously gained from this physiology division in UAS Bangalore has been the champion of champions when it comes to the Indian physiological scene. And this is something where the today's basic science is tomorrow's applied science, and I think they are contributing very significantly to today's basic science through their students and researchers, and I'm sure that this is going to be a wonderful opportunity for all of you to initiate. And I request Mahesh Sharapa also, by taking Dr. Nataraja Karaba's help, to initiate through postdoctoral research certain aspects where it is critically needed. With these few words, I thank Nataraja Karaba profusely for wonderfully taking us to the realms of the talk that you delivered today. Thank you very much.
Thank you.
Thank you. Thank you very much. Thank you.
Thank you.
Thank you so much, sir.
Thank
I
you.
really have to learn from you and Tiku Saab.